moving on to the last speaker for today, Dr. Ron Dixon from the University of Lincoln and Freedman Scientific Limited.
Congratulations, everyone. You've survived the day. It's been fascinating, busy. I'm Ron Dixon from Freedman Scientific, which is a consultancy, but in fact, I'm been associated with the University of Lincoln for about 20 years, and so now happily retired, but try to keep ex-PhD students and members of staff at Lincoln happily financed. So my speciality is grant applications. And we've been looking at the dynamics of AMR Campylobacter during the processing of chicken meat. And I'm very indebted to Bill, who's just given fantastic background and introduction. So I don't have to worry about that, and I've only got 4 minutes and 3 slides to do this in. So Campylobacter, as you might have guessed, remains important as the most frequent cause of food poisoning globally, with millions affected. The FSA is doing a great job in monitoring, particularly the AMR aspects, the transfer of drug resistance, in particular with the fluoroquinolones. So our study was to focus a little more than just looking at the retail aspect of meat production, but to look at the distribution of Campylobacter throughout from, as I say, from farm to fork. And an important aspect of from farm to fork is the abattoir. And so we started to look at, with our teams that you see here, funded by the Food Standards Agency in the UK, and with the University of Lincoln and Ferrous Science, one of the government-supported areas of science. And if you want to see more, and I won't have much time to tell you about it, this has been published in the FSA Research and Evidence under that website. So the evaluation of the persistence of AMR Campylobacter following the standard hygiene interventions in commercial settings. And I don't think many people realise that during the abattoir process, there are various processes involved to reduce pathogens, and usually involving steam, heating, cooling, and we want to see actually to what extent that affects the output of retail meat with Campylobacter. We also looked at E. coli, as you heard from Bill, as an important organism, but that's a little more complex, so we've left, this was a flash presentation, we've left that for another day. So to go to the methodology, there were 2 commercial poultry processing plants from a major English producer, and we looked at farm arrival from the farm, transported to the abattoir, and then the finished product. And we sampled 3 times on 3 occasions during the year, so if there was some aspect of seasonality, we might pick that up. And we sampled, and I say we, the team sampled both morning and afternoon. We were grateful that the main providers of this service were so helpful, and as an industry, they are concerned about AMR. So the first methodology we looked at was for the presence and abundance of Campylobacter. We looked very traditionally using Eucast guidelines at the antimicrobial susceptibility, first using disk diffusion and then the traditional MIC microtiter test. And then we looked at whole genome sequencing for all our isolates. And not on this slide, but we also looked at some metagenomics, which will be at the end. Okay. So the data is as you see it. I wonder if I can. Now, when I say we looked in the abattoir, we looked at every point that we could access during the abattoir process, from stunning pre-stun birds. And if you're not aware that these abattoirs with chickens stun the birds under very informed veterinary advice, and then they bleed them out, and then they go through defeathering, rehanging, inspecting, carcass inspection, washing. I know you can't see this in detail, and then a post-chill process where, again, through another steam and chilling process. So at the end, you notice Campylobacter abundance is quite low. At the beginning, it's quite high. And if you look at the farm, you will see that most animals come in. Of the 376 samples we looked at, 65.1% were positive for Campylobacter. So at the beginning, certainly, there's a high population in the UK farming community. We then looked at antibiotic resistance using the traditional MIC test. I don't know if you can see those numbers, but it shows that at each site, and these were site-specific, there were 2 sites involved, and either June or November testing. And, sorry, June, October, November testing, and they were specific as well. So we've got really quite a dynamic situation going on. 26% were resistant to ciprofloxacin, which is within normal limits of what we'd expect. 7% resistant to erythromycin, 72% resistant to tetracycline of our Campylobacters. We then did whole genome sequencing and found that the isolates were generally diverse. There's no real clear message on during the farm to fork sampling, but of the jejunii, we saw 5 sequence types, and you might recognize some of those as being endemic in some countries, and particularly 6175. It's 6175 being the most prevalent, and then most of those carried resistance genes to ampicillin or tetracycline or JAR-A. And we knew that because of our whole gene sequencing, but also we found that of the 137 subjects that we could look at diversity and ARGs, and interesting, and that, ooh, it's gone mad. Thanks. We're there. Yeah. So at the beginning, this is the crate stage, dirty crates as they arrive. We have high diversity in traditional metagenomic terms, and when we looked at antibiotic resistance, it was high. As we went through the process, it became less diverse and lower numbers of resistance. So it was readily recovered. Now, these metagenomic analyses are not related directly to the Campylobacter, but they are because Campylobacter needed special DNA preparation. But they give a message here of the hygienic provision was effective. So it was readily recovered from the process chain at early stages, but less so following industrial reduction measures, which I refer to as steam chill. And then the parallel sampling suggested that the hygienic practices were responsible for this. So when the FSA and others assay the dispatch, you can see that Campylobacter is very low. Or let's put organisms are very low. And so I think we've brought something, a deeper analysis to really quite a standard surveillance. And I'd like to thank you for that.